concepts and principles. So I have a privilege of introducing Dr. Anirban sir. He is principal scientist in the division of plant pathology, Indian Agriculture Research Institute, New Delhi. He has completed his graduation in agriculture in 1997 from BCKV, West Bengal. He completed his MSc and PhD program with specialization in the plant virology from this very division of plant pathology, ICAR-IARI, New Delhi. All along his academic career, he has maintained a very good record and received gold medal during his MSc and PhD programs. In his professional career, he joined as a scientist in Central Research Institute for Jute and Allied Fibers, Barakpur in 2003. Then in 2009, he joined as a senior scientist to NBPGR New Delhi. Presently, he is working as principal scientist, Division of Plant Pathology, ICAR-IARI, New Delhi. His area of research work is on virus diseases of plants. He had identified several new begomovirus infecting plants, jute, allied and fiber crops, pulse crops, vegetables and mustard. He developed the molecular diagnostics for detection of different begomoviruses. He had identified virus resistance germplasm of many crops. Currently, he has initiated the research work, which is a very important work, that is genome editing of viruses to development strategies for their management. With this research work, he has published 48 research papers in International Journal of Repute. In recognition of his significant work in the field of research, he has been awarded with many prestigious awards, among which the most prominent ones are Young Scientist Award from Indian National Science Academy, Indian Science Congress Association, Indian Council of Agriculture Research, and National Academy of Agriculture Sciences, which are worth to mention. Besides the research work, he is also involved in the teaching of postgraduate students for the Division of Plant Pathology. With this brief introduction, I welcome Anirban sir to deliver the lecture on molecular phylogeny concepts and principles. Please, sir.
Hello. Okay. Good morning, everybody. So low voice. I have not taken breakfast properly. So you have to be very much attentive. This is the first lecture, and it is my duty that I should encourage and infuse the enthusiasm of this entire training program so that it should go properly. And I am not an entomologist, but as a virologist, all the virologists are very much associated with the entomologist, because the viruses can only spread by the insect. So in that way, there is a very good rapport with a pathologist and an entomologist. Anyway, to begin the talk with, it is already, Sir actually told me to initiate the lecture at 11:30. It is already half an hour late. So I think you will be also, I'll drag you a little bit late in the afternoon, but there is no problem. This half an hour, if you see in the time scale of universe, is nothing. If you see at the time, 14 billion years ago, when there was no concept of time, only at the event which is called Big Bang, the time concept came. It is not coming, but it should come from here. So this is the event from where the concept of time started. And during that condition, there is a hostile condition. The environment is very much hostile. There was no possibility of withstanding life or other things, but in that condition also, different chemical molecules joined together to form the first biological form of this earth, that is a small RNA molecule. So it is not coming. So from that small RNA molecule, then the different more ingredient joined together to form a primitive life form, which is the primitive prokaryotes. And from this primitive prokaryotes, then different life forms evolved. And among those different life forms, we, this most civilized creature of our mother nature, the human being. Please, next please. So, these are the different life forms in the earth. Next, and with the civilized creature, the most intelligent creature of Mother Nature, the Mother Nature has created us, and now we are trying to take challenge with Mother Nature about our origin, how we have evolved. And the journey actually started from next the river bank to the Silicon Valley. The mysteries which has been created by Mother Nature, and it took billions of years to evolve these life forms in the earth. We are trying to solve that mystery of the Mother Nature by just sitting on a computer. Let us see how much we have progressed and whether we are really able to take challenge with the Mother Nature or not. Next. So, from the beginning of the is okay.
Just try it.
So, from the beginning of the life, always we have this kind of quest or not. Always we are trying to answer this question, what is life, how we evolved, how this we are always related to each other, and what is the element which carries all this information and passed to the next generation. So, to answer this question, different science evolved at different times. So, if you see the history, then there was a time when we call them as era of classical genetics. That time the people started think about the evolution theory and the principles of heredity, and during this time we are only thinking about the classical genetics part. Then there was considerable improvement in the subject of science. The DNA genetic material was established, extracrystallography, structure of DNA, all those things was established, and the triplet codon finally tried to say us that how this genetic information is passing, and it was known that DNA to RNA to protein the information is passed. So, this is the era of cellular molecular genetics, and then these three science subjects, classical genetics, molecular genetics and cell biology, they merged together to form the era of cellular molecular biology. So, you can see that how different times different science subjects they join together to give rise another new science subject. So, after this cellular molecular genetics era, there was a tremendous increase in the science or in the technological front. So, what happened actually? There are different technology like sequencing of the DNA, polymerase chain reaction, recombinant DNA technology. So, this technology then evolved, and during this phase what is happening actually, the classical genetics, molecular genetics, cell biology and technological advancement, they merge together to form the era of biotechnology. So, in that way again and again I am trying to say you that different science subject how they merge together to give rise another science subject. So, the biotechnology get momentum when the technological advancement was progress very much, very fast. So, what happened actually next? The sequencing technology in the early generation, when the sequencing technology was done by mainly two methods, Sanger chain termination sequencing and Maxam Gilbert sequencing, chemical sequencing method. During this phase we are trying to sequence individual gene, a portion of DNA like that, because the sequence speed is not that much, but in that speed instead of that, what happened during that phase, a very huge project was initiated, because we try to think that if we are able to know the code of ourself, what is written in our genome, then we will able to solve the mysteries. And during that phase what happened actually next, there was a big project on human genome sequencing, and it was very much surprising that before the estimated period which was thought that within 10 years we will be able to finish the human genome sequencing project, it was finished quite earlier. And then we could generate a first draft of human genome sequence. And after that, several more platforms of sequencing have evolved, like the next generation sequencing, the Illumina sequencing, Quagen sequencing, life technology sequencing, and Roche Pyro sequencer. So, these are the different sequencers. So, platforms, technological advancement by the engineers to help the biologist to solve the problems of the biology or to decode the genomes of many organisms. So, after that recently some more newer platforms have come, like single molecule real time sequencing based sequencing, next then nanopore based sequencing. So, all these sequencing methods, the basic principle is that how we can speed up the sequencing process and how we can reduce the cost of the sequencing. And when all these technologies came, what happens actually, the cost of sequencing is decreased significantly. As a result, the sequence number generated hugely. So, what happens if you see the growth of the databases, sequence databases, please see next. Starting from this 1995 onwards, there is a steep increase in the deposition of the sequence in the databases. So, when there is more number of sequence have been deposited in the database, now again we start asking question, okay, we have taken this challenge with the mother nature. We thought that if we are able to do the sequencing of the different organism, we will able to solve the mysteries. Are we really able to do that? Because after those sequencing, several questions came, like how these different organisms are different, because sequencing technology showed that all human beings are 99.99% similar. Similarly, the question like the function of the proteins, because ultimately we have to correlate the sequence information with the phenotypes. So, we will find out this function. Is it possible to find out the function of those sequences, how these sequences are related? So, this kind of questions started coming. So, people initially thought, when the human genome sequencing project came, people thought that okay, it is not a matter, a particular group of people which is having the advanced sequencing facility, they can solve this mystery, but it was not happening. So, they thought that okay, the entire university should be involved, that was not also happening. Then they thought okay, different universities should collaborate, that is not happening. So, again the history repeats. Next. So, actually the sequence, what happened actually, this is the sequence information generated like this. What is this? Anyone can give? Yes, this is hieroglyphs. Can you understand anything what is written here? No. Why? Why you cannot understand? Because you do not know what is the meaning of these things. So, something is written in code language. So, until and unless you decode it, you cannot understand what is written. Similarly, if you see a sequence, then sequence is just like a hieroglyphs, only the combination of 4 letters A, T, G, C. So, some information is hidden in this code language. Until and unless we decode this hidden information, we cannot able to understand what is written in this. And there is the challenge that one university, one person, one group, it is not possible to decode the information what is hidden. So, the history repeats again. The 4 major branch of science, biotechnology, information technology, computer science and statistics, they merge together to give rise the birth of a new subject called bioinformatics. So, it is a combination of different subject, definitely very much complex subject. It is not possible to decode the entire subject in one class, definitely, but I am trying to tell you a little bit about this bioinformatics and how that can be used to understand the genomics of whatever organism you are using. You may be using insect, virus, bacteria, fungus, anything, any living organism. To decode the information hidden in their genome, you need to apply the subject bioinformatics. And through my lecture, actually I am trying to give you the basics of those bioinformatic concept and principles. So, before going to the deep, first let me tell you that what bioinformatics is not, because that is the most important thing. It should not be a kind of subject or a kind of thing that you just for your good thesis or a beautification purpose you use this kind of things. So, you should understand that bioinformatics is not just using a computer to speed up biology, it is not just applying computer algorithms to biology, and it is not the accountant of the genomic data. So, what is then bioinformatics? The bioinformatics is the creative use of the computer to define and solve the central biological puzzles. So, I am trying to give you importance to this aspect, that it is a creative use of computer. So, it is not that computer will tell you anything, you have to tell computer what to do. Computer is a only hypothetic machine. If you guide the computer properly, then only computer will give you the answer. If you do not do that, computer can tell you anything. Now, it is up to you how you judge that. And second thing is that computer becomes hypothesis machine, making prediction which needs to be tested in the bench. So, just by prediction we cannot say anything, you have to now think by your own that whether this hypothesis prediction is correct or not. So, it is just a marriage of information science and biological science. Next. Just like the central dogma of biology, here is also a central dogma. This central dogma says, if there is a sequence similarity, there must be structural similarity. If there are two sequences, whether a nucleotide sequence or amino acid sequence, if you have two sequences which are matching with each other, then we must think that if the match sequence, match means whatever sequence is already available, and query sequence means the sequence which you have generated from your organism. So, if the query sequence is similar with the match sequence, then if the match sequence is having this kind of structural motifs like zinc finger motifs, then the query must also be predicted to be a zinc finger. Now, if there is a structural similarity between the two, then there must be functional similarity. So, if we know that zinc finger is present in the transcription factor and they have the function to bind the DNA, then our predicted sequence should also have the similar DNA binding ability. Okay. So, just by seeing a sequence, we can draw the function of that sequence. So, that is the beauty of bioinformatics, that it can reduce your search from several function of a protein. It can tell you that okay, this protein may have a DNA binding ability. Now, you have to test it in bench that whether really it is having a DNA binding ability or not. So, in that way we can find out the function of the protein by taking this kind of central dogma of bioinformatics. Next. So, bioinformatics actually have three basic components. The first one is development and implementation of tools and databases, which is not our domain, that is the domain of statistics people or computer science scientists. The second domain is development of new algorithm and statistical formulas, again the domain of statistics. And third one is use of tools to generate biological information and manage them. As a biologist, this is our domain. So, whatever softwares and algorithms has been generated, we will use them to solve the mysteries and to find out the solutions. So, on this basis actually this is my topic today, that sequence based molecular phylogeny, basic concept and terminologies. Because why I am trying to tell you, because many of you may be using this phylogenetic analysis and other things, but many of you may not have the clear concept or knowledge on the different terminologies which we are using. And as a result, what happened, that always I am seeing even the faculty students, they use those terminologies very irrelevantly, and they actually cause distract the actual ideas of this bioinformatics. So, let us start with the main lecture. So, suppose you are working with an insect. Now, you may work on a particular sequence of the insect, a small gene or a bigger entire genome or a kind of gene clusters and other things. So, anyway, you may have this kind of type of sequence information. You may have a small sequence, that is one gene sequence. You may have relatively big sequence like bigger genes, smaller genomes of viruses and other things. Big genomes means relatively bigger genomes of bacteria, and whole complex genome like eukaryotic genomes, insect genomes. So, they are big genome sequence data or whole genome sequence data. So, different types of sequence you may generate. So, if you have a small sequence information, the sequence may be 1 minute, when I will tell, come then. So, the sequence may be of small PCR product, only one read, that means just using a primer you are doing a sequencing, you generate the sequence, or you may have a relatively bigger gene, only using one primer you may not able to generate the sequence information. For that you may need sequencing of both the end and then generate the sequence of the gene. So, in any way what happened, if you have only one read, directly you will get the sequence, but if you have this kind of things that you may be next. So, you may be using two different primers, and in this case what happens, these two primers next they generate this two sequence which is not complete, but there may be a partial overlap region. So, how to generate a full sequence from these, what you have to do next. So, you have to take this sequence and do a reverse complementary sequence. That means, this sequence you have to take, you have to convert it into this sense sequence, and then next then join with the sequence, means just do a matching and then overlap you remove, and then this complete sequence you will get. So, in that way you first do a purification of the sequence and then you get your complete gene sequence. Next, you may also have this clone this gene in a vector, and in that case you can use the vector primer to do the sequencing, because if you use a primer, then you may not get the sequence of the ends, because the primer sequence will never come in the sequence. So, when you will clone them in a vector, in that case you will get the entire gene sequence, because in that case you are using a vector primer. But when you are using a vector primer to do sequencing, what happens? It generates a sequence which is contaminated with some vector sequence. Next. So, what happened? It will be like that a vector sequence contaminated part will be there, and then your gene sequence will be there. So, you have to remove this vector part by using different softwares. You can remove the vector sequence part and you can purify your sequence which of your interest. So, in that way the first step is that when you generate a sequence, you have to purify the sequence, you have to get the exact sequence. And remember that if you are working on insect diversity and other things, always try to take a sequence which is of same length. So, one particular isolate you may get 600 nucleotide sequence, another you may get 800 nucleotide sequence, another you may get 100,000 nucleotide sequence. So, you cannot analyze different length of sequence, because it will give you different result. So, always if you want to study a diversity, you have to take the sequence of the same gene and similar length. If the length is not same, the sequence result will vary. Next. So, in case of insect, what we generally use for sequencing for diversity analysis or characterization, these are the different genes like ribosomal small and large subunit sequences, internal transcript spacer sequence of rRNA, mitochondrial genes and nuclear genes. So, these are the different gene sequence which we use for individual gene or small part of the gene. So, these are the different example of different insect order, what are the different ribosomal sequences has been generated and people are using in that. Next. So, in case of mitochondrial gene, again there are several insect orders are there and different genes. The most important is cytochrome oxidase, Then these are the different gene sequence which people are using to characterize the insect or to analyze the diversity. Next, and there are a lot of 38 genes are there from the nuclear genes. So, those nuclear very standard genes are there that can be used to generate the sequence information for the insect. Next. So, these are coming under the one gene or small portion. Now, you may try to analyze a bigger sequence, not the whole genome, but a relatively bigger, a partial portion of the genome. In that case, the large DNA sequence can be fragmented and then sequenced by deep sequencing, which generates different sequence, and then you can overlap those sequence and find out this overlap region and remove this overlap portion to come out to a assembled sequence. So, in that way a larger sequence can be assembled and can be analyzed. Next. Now, if you want to do the whole genome sequence of insect, then three different terminologies came. Always you may encounter these terminologies. The first these three terminologies are reads, contigs and scaffolds. So, if anybody is working on the whole genome sequencing aspect, you may counter with this kind of terminologies. So, what are these things? So, whenever you will do the whole genome sequencing, the first thing is that you have to share the whole genome and you have to add adapter molecule to both the ends. This adapter molecule is actually a small DNA sequence which is known sequence and a primer is given. So, using those primers now you can sequence this unknown internal portion. So, after sequencing you get the reads. So, small sequence stretch which you will get, that is called reads. Then you overlap those sequence to join those reads, and then whatever you will get, this is called contig. So, different reads join together to form the contigs, and then from different portion of the genome such different contigs will be developed, and those contigs will then be assembled together to form a larger fragment which is called scaffold, and these scaffolds, different scaffolds join together to form the whole genome. So, in that way, in case of whole genome, starting from the reads to contigs to scaffold, ultimately we are able to generate the whole genome sequencing data. Next. So, there are several softwares which are available for assembling those reads and contigs. These are the contigs assembly programs which can be used to align and to develop the contigs. Next. So, after doing the sequencing, what the company people will give you is like this kind of sequence. So, it is a kind of chromatogram which shows you in different color different nucleotides. So, A, T, G, C, these 4 nucleotides are denoted in 4 different colors, and these are the sequence actually generated by the company, and this kind of file is called the trace file of the sequence, and this trace file very common format is .abi, just like .doc, .excel or .ppt. Similarly, for the sequencing there are different formats are available. The company people which will give you, they will give you in .abi format or any other formats that I will tell when I will show you the practical. So, this kind of information they will generate, or if it is a whole genome sequencing data, they will be presented like that. So, whole genome sequence information. So, this is the raw sequence which you get. Next. So, after getting the raw sequence from the company, the next thing is that to find out the meaning of the information which is hidden in the sequence, and for that we need to compare the sequence with other sequence. Without comparing, we cannot able to know what is hidden in that, and from where we get those other sequence? We get those other sequence from sequence databases. So, there are several sequence databases. Next, those can be used to generate the or to compare the sequence which you have generated, and these biological databases, they mainly have 3 different components. The first component is update component. Whatever sequence databases are available, you need to update that. It is not a closed database that you cannot update it, if then there is no meaning. So, wherever in the world, whoever is working on sequencing, if they can generate a sequence, they can upload their sequence in the database, and then that means itself you can update the sequence. It is not that some person will be there, you have to send to them, they will upload the sequence. You can update the sequence database, but there must be some administrator to check your validity of the sequence. Then second component is query component, so that you can ask the database that I want this kind of sequence, show me those sequences, because in the database the sequence of human genome, sequence of mice genome, sequence of rice genome, sequence of insect, sequence of bacteria, everything is deposited. So, if you have to find out the sequence of your interest, then you need some tool to search, and that is the query component. When I will show you the practical, I will show you how you will ask the database to for your query that you show me these sequences which of your interest. And third aspect is retrieve component. You not only you want to see the sequence in the database, you want to take down those sequence into your computer for their analysis. So, there must be a retrieval component, so that you can take out those sequence from the database. Next. So, these biological databases are mainly of two type. One is sequence database and second one is called structural database. So, sequence database again divided into two part, one is called nucleotide sequence database and second one is protein sequence database, and the structural database is structural database is PDB, the protein database where different structures of protein have been deposited. Now, in the nucleotide sequence database there are three major players: NCBI, that is National Center for Biotechnology Information from USA, EMBL, European Molecular Biology Laboratory from European countries, and DDBJ, DNA Data Bank of Japan. So, these are the three major players for the sequence database. Whatever sequence you deposit in the database, you have to deposit in these three databases. Now, the question is, suppose if you have generated a sequence, where you will submit, in NCBI or EMBL or DDBJ? You can submit the sequence in any sequence database. There is an agreement between these three major players that whatever sequence has been deposited in one database within 24 hours, that will be circulated to other databases. So, anywhere with any database you can deposit and you can search your query sequence from any database. So, there is no restriction for that, but besides this important three major players, there are several specialized sequence database like human genome sequence database, mice genome sequence database or rice genome sequence database, but all those sequence information which are there in the specialized sequence database, they are also present in this sequence database. It is not that the specialized sequence database is different from this specialized sequence database only to analyze the sequence more with those sequences only, for that those specialized sequence databases have been created. Now, in the protein sequence database there are SwissProt and TREMBL, that means translated EMBL. So, these are the protein sequence database which has been, you can deposit your protein sequence, amino acid sequence in these databases. Next. So, these are the some screenshot, this is the NCBI database, next EMBL database, next DDBJ database, SwissProt and TREMBL, this is the structural database PDB. So, these are different databases, some specialized sequence database like Varicella sativa database, then some fungal sequence database like that. So, there are several such kind of specialized sequence databases are also there. Now, how to deposit these sequences which you have generated into the database? Next question is that you have a sequence, how to deposit? Now, for sequencing there is a option called GenBank, or NCBI is having this option which is called GenBank, not GenBank. So, in GenBank there are two options, one is called Bankit, another one is called CQIN. Bankit is an option if you have only one sequence or few sequence of a gene you want to submit, then you use Bankit. But If you have a cDNA library of your insect, then you cannot submit it through Bankit, you have to submit it through Sequin. So, if you have a large sequence database, then you submit it through Sequin option of GenBank, and if you have few gene, then you submit through Bankit. Where these are located, we will show in the practical that how to submit a sequence through Bankit and Sequin, that time it will be clear, but this class is only for understanding you with these terminologies. And in EMBL there is another option called Webin Align. So, there is an option, a portal, through this portal you can submit your sequence in EMBL database. So, these are the different portals through which whatever sequence you have to deposit, and remember that whenever if you are working with a sequence, it is your duty to submit the sequence in the database. Without submission of the sequence, your sequence has no validity, and whenever you will sequence your whenever you deposit your sequence, the sequence database will give you a number which is called accession number. So, that accession number is a kind of IPR for you, that I have developed a sequence or generate a sequence, and this is my accession number. Nobody can claim that accession number for them. They can use your accession number for their analysis purpose, but that accession number is purely belongs to you. So, there is no question that if I will generate a sequence, somebody will steal that sequence and they can do for there. They cannot do, because they will give a accession number and that is for you only. So, next. So, this is the option Bankit in the GenBank, where you can submit the sequence. Next, and this is the Sequin, again in the NCBI database there is the option of Sequin to submit large volume of sequence. Next, and this is the, if you want to submit the entire genome and other things, then also there is option for submission of the genome sequencing database. Next. So, before submission of the sequence, you may heard this term that you need to annotate your sequence. So, what do you mean by annotation? Annotation refers to commentary or explanation of the information appended to a DNA or protein sequence stored in the database. So, if only you submit a sequence just as ATGC, nobody will understand what is this. So, you have to give a commentary that this sequence belongs to this, and these are the different isolates. From this country it has been isolated. So, if you are not append those sequence in the along with your sequence has no meaning. So, whenever you try to submit a sequence, try to give as much as annotation as possible, then your sequence has much more visibility to others that this sequence is a full annotated sequence. So, what are the kind of different annotation you can make? Like source, from where you are isolating, which insect, which country or which organism, then what is the protein sequence of that, what is the predicted protein structure, if you can give the different domains of the protein, quaternary structure of the protein, protein functions, so and so forth. So, different options are there. So, you have to append along your sequence those information, and then only you can submit. So, that means whenever you generate a sequence, it is not that immediately you submit a sequence, you have to annotate that, and for annotation you need to know all those things, and that is why you need to analyze your sequence first, and when you analyze, then you can come to know that what are the annotation you can append along with your sequence. Next. So, you sequence a genome or gene of an organism and you want to deposit it in the database. After that, what you have to do next? So, the first thing is that you need to analyze the sequence, and for that you need to get the sequence, other sequence from the database. So, if you have your own sequence, but you want to analyze that sequence, you need to compare the sequence with other sequence, and that sequence it is already deposited in those sequence databases. You want to take out those sequence, and that is the terminology used for that. the terminology is called data retrieval. It is the collection of the data from the database. So, as you are listening this lecture, I hope from your any seminar, any presentation, any thesis work, anywhere, you never use the term download. Okay? You cannot download a sequence from database. You can retrieve a sequence from the database. So, what is the difference between download and retrieval? Just I am giving you one example. Suppose if you have a Word file, everybody is using Word file. So, in that Word file, suppose you have 10 pages. If I will tell you download the third page of the file, can you download? You can download the entire file. You cannot download a single page from a file. You can copy it, paste it in another file, but you cannot download a single page from a file. Similarly, the databases are actually a file and the sequence information are individual pages of that file. Okay? So, never sequence can be downloaded, it can only be retrieved. Okay? So, from onwards, now this day onward, you should not tell that I have downloaded 10 sequence. No, you have retrieved 10 sequence. Okay? So, why we need to retrieve sequence? Next, to analyze two basic things. One is called genomics, one is called proteomics. So, what is genomics? It is understanding the interaction among the genes, and proteomics means understanding the interaction among the proteins. So, in the genomics there are again three different things. One is called structural genomics, which deals with the genome organization and ORF prediction or gene prediction, designing of the primers, and in comparative genomics the ORF annotation, relationship and variability between different pathogens or insect and their strains, and the molecular phylogeny, which comes under the comparative genomics. And third one next is the functional genomics, that means prediction of the motifs, block and function of the protein, this comes under the functional genomics. So, under the genomics there are three components. If you retrieve a sequence, you can analyze the genomics, which is the topic of the training program. So, you can do the structural genomics, comparative genomics or functional genomics. Also you can do the understanding the function of the protein next, which deals with the interaction of the proteins with other molecules and drug designing, docking, those kind of things comes under the proteomics. So, this type of things you can do if you retrieve a sequence from the database and you analyze those retrieved sequence with your sequence of interest. Next, the next terminology which comes is data mining. So, one is data retrieval, that means collection of the data from the database. Second one is called data mining. Data mining is the generation of the information from the data which you have retrieved from the database. So, now you want to generate information. So, that is called data mining. So, data mining are of different types like molecular variability and phylogeny analysis, gene finding, primer designing, protein structure and function, promoter identification, so on and so forth. So, all those things comes under the data mining. Today's lecture I will mainly concentrate on this molecular phylogeny, how you can do molecular phylogeny using the bioinformatics. Next. So, still now lot of talk I have done, just a little bit relaxed time. Just tell me what is this? Everybody understood, it is bullet. I do not know how you have understood. Have you seen any bullet, anybody? No, but you have understood. Why? Because you see this picture. Next, this is pistol. So, this pistol is throwing that bullet. Next, again bullet. Next, what is this? What? Tell. No, it is the nozzle of the pistol. So, pistol nozzle is having this kind of different spiral things. So, when a bullet comes out from the pistol, it is not straight, it is already rotating, and that is why pistol, when hit a bullet, it passes through a small hole, and when it comes out, it gives a big hole because it is rotating always. Okay. So, this is the nozzle, and this nozzle has this kind of grooves inside it. So, when a bullet is thrown from the pistol, what is happening? The bullet is getting scratched with these grooves which is present inside the nozzle. So, what happens next? So, this is kind of scratch mark which the bullet acquire when it throws from a nozzle of a pistol. So, next. So, what happens when a bullet passed? So, it creates some scar mark in the bullet, and the bullet also injures actually the nozzle. The second bullet when you throw, it also carries the earlier scar mark and some new scar mark created in the bullet. Next. So, next bullet, again all those scar marks which has been previously there along with some new scar mark comes. Next. So, in that way, whatever bullet is throwing, you are throwing, they have some scar mark which is common to all, and that is the detectives actually try to find out that whether this bullet or few bullets what are there in a crime scene, whether they throw from the same pistol or not. How they find out? By analyzing this scar mark. If this scar mark has some kind of similarity with the nozzle scar mark, then they throw from the same nozzle or same pistol. Okay. So, if you think that thing in the biological science. Next. So, this is the process of evolution through these different mutations created in the genome, and this is the variation in the individual. But though there is variation, by tracking the similarity among those individual with the process of evolution, we can know that whether they are related or not. Okay. So, by taking this analogy, what I mean to say that whenever any process of evolution taking place, mutation taking place, new progenies are coming out, new strains of insects are coming out, they must have some kind of similarity with their original parent through the evolution process, though they are different in the sequence, but there must be some similarity among those sequence, and it is our duty to find out whether that similarities exist or not. If they are exist, then we can say yes, these individuals are related to each other. Okay. Next. So, what is then phylogeny? Phylogenetics is the science of estimating and analyzing this evolutionary relationship. So, whatever evolutionary relationship different individuals have, that can be analyzed or understood by the understanding of this phylogenetics. How it is done? It is done through analysis of the DNA sequence, which helps to understand the phylogeny of an organism. And what is the purpose? The phylogenetic relationship helps us to understand the evolutionary history of an organism and how they are related to each other. So, this is the purpose to understand the molecular phylogeny. Next. So, what we want to know from the molecular phylogeny? What is the basic things that we want to know? We want to know three basic questions we want to understand. The first, whether our sequence has any homology with other sequence, how much similarity is there between the sequence which we have generated and the sequence which has already been deposited, how much is the similarity, and how our sequence is phylogenetically related to other sequence. So, these three basic questions we want to know from the phylogenetic analysis. Now, these three terminologies came: homology, similarity, and phylogenetic relationship. So, one by one I am trying to explain what are these three terminologies. What do you mean by homology? Many persons know these things. What do you mean by homology? Homology is a state of gene or morphological characters that shares a common ancestry with different gene or morphological character.
So, it is a state of gene or morphological characters, and there must be a common ancestral relationship. If there is no ancestral relationship, we cannot call them homologous sequence. So, homologous sequence must have ancestral relationship and they must have high level of similarity. So, if you think about the sequence information data, so in sequence information data, it is taken to mean that two sequences or even two characters within the sequences are descendant from a common ancestor. Two sequences are descendant from a common ancestor, then only we call them as homologous sequence. Now, here is the question. I have seen that almost all the students have this misconception of the homology, because almost all of you in your seminar or anywhere you may mention this kind of things. You may misuse or abuse the term homology with similar, because this kind of statement I have heard many times, like two sequences were 70% homologous. Many big scientists are also telling like that, 80% homologous sequence, 20% homologous sequence, which is wrong. Sequences show a certain amount of similarity. From this similarity value, we can probably infer that sequences are homologous or not. Okay? Why? If you, again some lighter joke I would like to tell, a famous quotation of Walter Fitch next about the homology. He defined homology is like pregnancy. You can either pregnant or not. Can anybody tells whether a woman is 20% pregnant? No. We can only tell a woman is pregnant or not. We cannot tell 70% pregnant, no, because pregnancy is a qualitative term. It is either pregnant or not. Similarly, homology is a state, is a condition, is a qualitative character. We can only tell whether two sequences are homologous or not. We cannot tell 20%, 70% homologous, no. So, never misuse this term. If you are talking about homology, you only directly tell two sequences are homologous or not. You cannot tell 70%, 80% homologous. Clear? So, next. So, homology cannot be measured, only we can say whether homology is there or not, but we can measure similarity. And from this similarity value, we can predict whether two sequences are homologous or not. And remember, another thing is that homologous sequence must have similarity, but if there is similarity, we cannot say there is homology, okay? Because homology is related to ancestral relationship. If there is no ancestral relationship, we cannot tell the sequences are homologous. There may be similarity, but we cannot tell. If there is similarity along with the ancestral relationship, then only we can tell the sequences are homologous. Okay? Next. So, homologous gene superfamilies are of two types. This when we are talking about homology, homologous gene families are two types. One is called orthologous gene, another one is called paralogous gene. What do you mean by orthologous gene? Now, like example like DNA polymerase of goat, DNA polymerase of human. So, these are same function, same kind of sequence, but they belong to different taxa. So, in that case we call them orthologous gene, and they arise due to the result of lineage transfer. So, during the process of evolution, when there is a lineage transfer between different species, then this kind of orthologous genes have been created. And the other term is paralogous gene, the example like hemoglobin and myoglobin. So, they found in same taxa similar sequence, but they have different functions. So, in that case we call them as paralogous gene. So, when you work on any insect, on any living organism, you come across with that your insect, suppose a whitefly gene sequence have also be present in the aphid. So, there are two different things, but they are present, that means that is a kind of orthologous gene. But there may be some gene, the domain, same domain is present repetitively in the whitefly genome itself, that means those sequences are paralogous genes. So, whenever you analyze a sequence, you have to think on that whether I am trying to find out a orthologous homology or I am to find out a paralogous homology. What kind of homology we want to understand, because I repeatedly I am telling you, it is the creative use of computer, you have to dictate the computer what you want. Otherwise, whether it is orthologous, whether it is paralogous, whether whatever thing you give a sequence to the database or software will give you some result, and then you cannot interpret that result because you are not directing the computer what is your query. So, you have to direct the computer what kind of things you want. Next. So, these paralogous genes are developed due to gene duplication during the process of evolution. So, what is our main goal? As I have told, from a sequence our target is to find out homology, but directly we cannot measure homology. Then how we want to measure homology? To measure homology, we have to measure similarity or identity. Again, too confusing term. Okay? We misuse this terminology very much. Similarity, identity, there are two terms, different things. So, what do you mean by similarity? What do you mean by identity? So, next. So, if you see these two sequences I have written here, this is one second. Next, what happened? In this sequence, the first nucleotide and second nucleotide is A and T, here is also AT, last nucleotide is AA, remaining nucleotide is not matching. Okay? So, this matching which is there is exactly matching, or we call them as identical match. So, this is called identity, when that there is exact matching of that nucleotide in the same position of another sequence. Next. Now, you see these two nucleotides, C here it is T or A and G. So, here C means pyrimidine is replaced by another pyrimidine, a purine is replaced by another purine. So, as a chemical composition, purine is replaced by purine, pyrimidine is replaced by pyrimidine. In that case, we call them as similar match. This is also similar and this is also similar, but if we call only identity, that means only identical match. So, similarity includes both identity and similarity, but identity is only identical sequence. Okay? Next. Now, this sequence you see, a C is replaced by a pyrimidine is replaced by a purine. So, in this case it is called mismatch. Okay? So, there are three things, one is identical match, similar match and mismatch. Okay? Now, how we are actually comparing this sequence, these two sequence, the first nucleotide with first nucleotide, second with second, like that. So, we are taking the two sequence up and down and trying to understand the positional similarity or identity. What is there in the first position, same thing is there or not. We are trying to find out the positional similarity, and this positional similarity or identity is called alignment. So, this terminology often you use, that we will do a sequence alignment. What is alignment? Next, alignment is an computational hypothesis which identify positional similarity or identity between the bases or amino acids. So, it is the hypothesis which states you the positional similarity and identity between the bases and amino acids. So, just like this. Now, this alignment may be done by two ways. You can align, next, you can align through global alignment, you can align through local alignment. Next. So, what is this global alignment and local alignment? So, if you try to align, this is your target sequence which is deposited in the database, and this sequence you have generated. So, your sequence is partially matching in one stretch with the sequence which is deposited in the database. So, the software will not analyze the entire sequence of the database with your sequence, because in your sequence only a stretch of sequence is matching with the sequence which is deposited. So, if that kind of analysis has to be done, that a stretch of sequence which is showing high similarity or identity, that is analyzed by the software, then that kind of analysis or alignment is called local alignment. Now, if the alignment is done from the first nucleotide to the last nucleotide, then maybe many mismatch or many thing is missing, but it has to be done from the first to the last, then that kind of alignment is called global alignment. So, through global alignment we can exactly know what is the percent similarity between or identity between the two sequences, but through local alignment we cannot do that thing, because it is only searching a stretch of sequence which is showing high similarity value or high identity value. So, through local alignment we cannot find out the exact percentage of identity or similarity, but through global alignment we can do that thing. Now, to do this alignment, this kind of alignment, we have several softwares which we use. Next, before doing that thing, first I would like to tell you that how actually this alignment are done. Suppose we have this sequence, one 5 nucleotide sequence, one 7 nucleotide sequence, and there is mismatch 2, then match 2, these two are matched, 3 mismatch, and there are two gaps. So, using this sequence, how we can align? We can align like that, then there will be only mismatch 2, 3 mismatch and 2 gaps. So, our aim is to increase this match. So, we can use these 2 gaps in that way, like you can put this one gap here and one here. So, now you can see the match is increased, okay, and mismatch is decreased. Utilizing the similar gaps we can do this. Next, now we can do like that these 2 gaps we put this together. Now you can see the match is 5, mismatch is 0, gap is 2, okay. So, only these 2 sequences, if you compare, this is the most important alignment, because here we are increasing the match, and for that what we are doing? We are doing some mischievous thing. We are creating some gaps in the sequence. That kind of gap is not there. For doing our purpose only, we are creating those gaps in the sequence. As we are doing that thing, so we must have to be penalized for that, okay. So, whenever you put a gap in a sequence, the value of this alignment or the score of this alignment, suppose if I will give the score 5 here, because 5 mismatch, 5 if I will give score here, but as I have introduced 2 gaps here, I have to remove 2 marks from that, because that is not the ethical thing I am doing. So, 2 marks penalty I have to give, and then only the score will come as 5 minus 2 as 3, okay. So, whenever you introduce a gap, that is called open gap, okay, and that open gap whenever you introduce, that gives you a penalty, you have to give a penalty for that. And after this open gap, then you introduce another gap, that is called extended gap. So, for extended gap also you have to give a penalty. That penalty is little bit smaller than the open gap penalty. So, whenever in the sequence you have to introduce a gap to get a proper alignment, you have to give some penalty. So, the entire score value for that alignment will reduce, okay. So, in that way we can do next. So, this is called open gap and this is the extended gap, and this for these 2 gaps you have to give some penalty. When I will show you the practical, that time I will show you what is those open gap penalty and extended gap penalty. Softwares will determine that thing, you do not have to bother about that, but how the software is actually working beyond the screen, so that actually I am showing you. So, this kind of huge thing, they have to do permutation and combination, and they have to give you the alignment. It is not possible by if the sequence is very large. Only by 2 sequence I am doing this 3 different combination. So, if the sequence is so many and so many large sequence, 1000 nucleotide sequence and 10-15 sequences are there, how to do next? So, for that next we have to do dynamic algorithm of programming, and that is where the statistics people and computer scientists helps us to develop this algorithm or programming, so that this permutation and combination manually we do not have to do, the software will do for us, and when I will give the 2 sequence, software will do this permutation and combination and give you the exact alignment which is having the high score after even reduction of the penalties and other things. So, programming, after the programming it generates some scores. So, any programming when they try to align the sequences, after alignment they generate a score. Score means some points for that alignment, that is called score, and also generate E values. E values means probability of not matching the sequence with other sequence. So, it is a statistical jargon. E value means probability of not matching the sequence with other sequence. So, when we will do the practical, that time we will see that if the E value is 10 to the power minus 3, that means the probability of not matching the sequence is 0.0001. So, if probability of not matching is 0.0001 means probability of matching is 99.9999. Okay. So, the 2 sequences are highly similar. So, high score value and low E value means 2 sequences are highly similar or identical to each other. Okay. Next. So, this is a kind of score matrix which the software creates. You do not have to remember or note anything. Softwares in background they create this kind of score value. So, this is ABCD is actually the sequence which you have deposited. This is another sequence. So, between these two, this is the score value, between this and this is the score value. So, in that way they create a matrix table of the score value and they will tell you what is the relationship between different sequence, what percent of similarities are there. Next. So, sequence alignment, what I am talking about, that can be done by different softwares, BLAST, FASTA, Bleach, Beauty, ClustalW, Mega. So, these are the different basic softwares. There are many others high end softwares, but I am talking about very basic softwares which will show particularly 2 softwares, this BLAST and Mega, to make you understand that how this alignment can be done. So, the first thing is BLAST. Next. So, BLAST is not bomb blast. Again I am telling many students, many faculty have seen, they again misuse this term. They tell that I have blasted the sequence. It is not bomb blast. So, blast is not a verb that you will tell blasted the sequence. BLAST is an abbreviated form of basic local alignment search tool. So, it is a local alignment, as I have told you, it is a local alignment search tool. Using this tool you can analyze your sequence. So, never in your life you will tell that I have blasted the sequence, you will tell I have analyzed the sequence using BLAST. Okay. So, these terminologies you remember, do not use those kind of unnecessary things. So, BLAST is the algorithm used by a family of 5 programs that will align your query sequence against a sequence in a molecular database. Next. The statistical methods are applied to judge the significance of the matches, and alignments are reported in order of this significance as estimated by the applied statistics. So, they will do this, BLAST will analyze your sequence, query sequence with all the sequence which is deposited in the database. So, millions, billions of sequence deposited in the database, the software will analyze that thing and they will tell as per the significance based on the score value and E value, they will tell you this sequence is having highest score value. That means, with your query sequence this sequence is showing highest similarity value. Okay. Next. So, BLAST actually have 4 different things, 4 different analysis you can use through BLAST. The first one is BLAST N. So, BLAST N is the module in the BLAST which compares a nucleotide query sequence against another nucleotide query sequence. So, if you have a nucleotide query, you can analyze with another nucleotide sequence. Okay. BLAST P compares amino acid query sequence with amino acid sequence. Next, BLAST X. Suppose you have a nucleotide sequence, insect nucleotide sequence. You want to know that with which protein your sequence is matching. You do not have any protein sequence, but you want to know the sequence which I have generated, whether that is having any protein coding sequence or not. For that, you compare the 6' computational translation product of this nucleotide query sequence against the protein sequence database. So, they will analyze the nucleotide sequence, first convert them into 6' protein coding sequence from both the strands, and then compare those deduced amino acid sequence with the protein sequence database, and tells you that your nucleotide query sequence may code for this kind of protein. Okay. So, if you have a nucleotide sequence, directly you can able to know that what kind of protein it may codes. Next, now you may have a amino acid sequence, you may not have nucleotide sequence, you may have a amino acid sequence. You want to know that this particular protein is coded by which gene of the insect. I have a toxin protein, that toxin protein is produced by an insect gut. I want to know that which particular gene is actually coding for that protein. For that, you have to use T-BLAST-N. So, T-BLAST-N is another module in the BLAST through which you can analyze this. Next. So, this is the after the analysis you will see the BLAST in this way. You have to give your query sequence here, and after that it generates the score here. So, this is the score value, E value. Whenever we will show you the practical, that time you will be able to see it more clearly. Next. So, what we know from the BLAST? Next, we know from the BLAST that sequence that shares similarity with query sequence. Only we are able to know the sequences which share similarity with your sequence and helps to retrieve those sequence. Through BLAST analysis only we can able to do only up to that. Next, but we cannot know from the BLAST, we cannot quantify the sequence similarity or identity because it is a local alignment. Local alignment, again it means only a stretch of sequence which is showing highest similarity region, that only it is analyzing, it is not analyzing from start to end. Okay. So, that is why you cannot take the value, though the BLAST will give you a value percent similarity, but you cannot take that thing, and it cannot tell about the phylogenetic relationship. The phylogenetic relationship cannot be understood from the BLAST directly, you have to do some other analysis for that. Next. So, why I am talking about that you cannot understand or cannot tell the percent similarity, because here I have taken one example from the BLAST analysis. This is your query sequence, this is the subject sequence or the sequence which has been deposited. Between these two sequence you can see 373 is the query sequence nucleotide. Out of 373, 346 sequence they are comparing, not the 373, and out of 373, 346 sequence is matching, and that is showing 92% similarity. Okay. So, next. So, it is like that this is your deposited sequence and your query sequence is matching like this, small, not the entire things. So, it is 92%. Now, again you go down to that analysis, you may see this kind of things that your query sequence is matching with another sequence, and in that case out of 373 it is only comparing 84, and out of 84 77 is matching. So, here it is showing the similarity value 91%. So, in that case it is comparing 373 and 346, out of 373 is matching, it is 92%. Here is it is matching, out of 84 77 is matching, it is also showing 91. That means these two sequences are same, similar kind of things? No, because in this case only 84 nucleotides. So, next. So, in the entire thing only this much portion is matching, not the entire portion is matching, but it is showing you because it is comparing only this region. In this region it is showing 91%, it is not comparing with the entire thing. That is why from the BLAST value we cannot directly take percent similarity value, though it is giving a percent similarity value in the column, you should not take that as a percent similarity value. Next. So, after the blast, if we want to know that how these sequences, how much similarity is there and how much percent similarity is there, then we have to do multiple global alignment. So, multiple global alignment means you have to take the global alignment and not only one or two sequence, with many sequences you have to take. And in this case, first we have to do a quick pairwise alignment. For individual pair, we have to take quick pairwise alignment. Suppose you are taking 5 sequences, so it will analyze 1 to 2, 1 to 3, 1 to 4, again 2 to 3, 2 to 4, 2 to 5, like that, all the combinations of pairwise, and from that pairwise alignment it calculate a matrix, and that from that matrix value first it develop a guide tree. Guide tree means a relationship tree, that if these two sequences showing highest value, they are coming together, then it takes that two as one group and analyze the sequence with other sequence and finalize it that with which sequence that group is matching. So, when again I will do the practical, that time this kind of things will be much more clear, but at least understand this terminologies, that we will have to develop a guide tree, and from that guide tree it will do a progressive alignment, and then it will generate a final relationship value. Next. So, this kind of multiple alignment we will be able to do, and then we can generate this kind of percent similarity matrix or percent identity matrix. So, here I have taken one value of different isolates of a virus, and these are different values of the percent similarity, and then next, this is the tree or relationship tree has been developed, which showed that the particular isolate is belong to which particular group or which particular cluster, and this type of tree is actually the phylogenetic tree through which we can analyze the phylogenetic relationship. Next. So, what is a phylogenetic tree? It looks like this kind of things. Okay. So, there is a root from where it originates. Okay. So, then this there is a kind of junction, and this junction, these and this kind of junctions are called nodes. They represent a taxonomic unit. So, different nodes indicate a particular taxonomic unit. These are called branches. These branches represent the relationship. And next, if two branches are come together under one node, two branches are coming together, they are called sister taxa. So, they are closely related to each other under this same taxa. Next, and this is the length of the branch. This length of the branch indicates the genetic distance, that how much evolution, how long years evolution has taken place, that is denoted by this length of the branch. But not always we are using this length, because many times we are not directly looking at the evolutionary perspective. So, and another thing we may have seen, that there is some number value which is present at the just at the below the nodes, and this value is called bootstrap value, or this bootstrap value tells the significance that these two branches needs to come together. Okay. If this value is high, that means these two branch always comes together. If this value is less, that means this branch, the probability of these two branch to come together is very less. Next. So, the branching pattern of the tree is called the topology of the tree, and the representation of the relationship through the line is called dendrogram. So, whether it is a phylogenetic tree and dendrogram, they are the same terminology, similar things, both depict the representation of phylogenetic relationship through lines, that is why they are called dendrogram. Next. These dendrogram are of two types, one is called phylogram and another one is called cladogram. So, these dendrograms, the representation of the relationship is may be presented in two ways, through phylogram or through cladogram. and they can also be represented, both the phylogram and cladogram, they can be represented by a rooted tree and unrooted tree. So, again 4 new terminologies came: phylogram, cladogram, a rooted tree and unrooted tree. What are them? So, first come to phylogram. What is phylogram? Phylogram is a phylogenetic tree that indicates the relationship between the taxa and also conveys a sense of time or rate of evolution. So, it is not only grouping your taxa, it also tells you about some time. This temporal aspect of the phylogram is missing from the cladogram. Okay. So, how they looks like next? This kind of dendrogram, they looks like this. If anybody has any time used this software to generate this kind of tree, you may see some trees look like this, that some branches goes here, some are goes not in the same line, some are much more branched. So, this kind of phylogenetic tree, if anybody have used, they may come across with this type of tree. So, this kind of tree is called phylogram. Why? Because this length here indicates the time. Okay? So, the length of the branch indicates the time. So, a temporal concept is there. Next. So, this type of branch which is there, which is going ahead of other branches, it indicates this branch or this clade are evolutionarily more advanced. In this case, you just see here, if this is the length of time or the time intervals, then after few years or million years or something, they separated out. But here they are going on, and after long time they separated out into 2 different branches. Okay? So, these branches are evolutionarily more conserved. Here they are evolutionarily more advanced, and in this case next, they are evolutionarily much more diverse, because there are more changes. So many branches are there in one particular node. That means they are evolutionarily much more diverse. So, if you want to analyze your data based on this kind of interpretation, it depends upon you. What kind of data you have and what kind of analysis you want to do, that depends upon you. It is not that software will tell you whether you have to develop a phylogram or whether you have to develop a cladogram. That is you have to dictate the computer, the software, that whether you need a phylogram or whether you need a cladogram. If your data do not convey a sense of time, never use phylogram. Why? If you use the phylogram, then definitely the paper, if you publish, the reviewer will ask you that why you have generated this type of tree phylogram, because you are not analyzing anything based on the evolutionary consequence. Okay. So, if your data is not related to evolutionary perspective, then you should not use phylogram. This is not for beautification. I am telling you, many students, many people have seen गोल tree banata hai, so many things they have prepared, that is not required. What for? Because your thing is not for beautification purpose, it is to, you are representing your raw data in a visual form. Okay. So, there is no need to do any beautification. The cladogram, on the other hand, it is the same thing, but the temporal concept is missing. So, if the temporal concept is missing, that means the length of this tree is same. So, next. So, all these branches ends at same point, there is nothing is going ahead or going beyond. Okay. So, everything ends at same point. That kind of tree is called cladogram or rectangular cladogram or phenogram. The similar cladogram can be presented in this way, which is called slanted cladogram. So, in the software all these options are available. How, what is your requirement? You have to ask the computer, I want a slanted cladogram or I want a rectangular cladogram. So, what are the different purpose for this different type of trees? So, rectangular cladogram, that indicates that when you want to do grouping for taxonomic studies, you want to do a taxonomic studies, you want to do a variability studies, in those cases you only use rectangular cladogram or phenogram, do not use any other phylogenetic tree. Now, in slanted cladogram, when we want to know convergence, divergence, parallelism in the evolutionary studies, then we use slanted cladogram. So, again it is related to evolutionary things, but not with time things. So, what is this means? Convergence, divergence, parallelism. Next. So, you see this is the convergence. This indicates that you can predict that after few million years they may join together, they may arise from two different origins, but they may join together. These are the parallelism, that their evolution is parallel evolution, nobody is influencing any entity. So, their evolution is parallel evolution. And next, these are the divergent, that upon going on in the evolutionary process, after some time they diverged out from the main branch and they creates another taxa. So, if you want to analyze your sequence based on this kind of interpretation, you use slanted cladogram. Otherwise, if you are only using for your taxonomic purpose, for your diversity analysis purpose, only use a rectangular cladogram, no other things. Next. Then another two things, rooted dendrogram and unrooted dendrogram. Next. So, if there is a root, then you take this rooted. If there is no root, you do this unrooted. So, what is root and unroot? Suppose you want to analyze a cytochrome oxidase gene from whitefly, okay. Different isolates of whitefly cytochrome oxidase gene. Definitely it is assumed that all this cytochrome oxidase gene from different isolates of whitefly is actually originating from a whitefly, okay. So, in that case you think that there must be a root, but if you take one cytochrome oxidase gene, another gene from another aphid, another gene from another insect, different things you try to analyze, in that case you cannot think that all those things coming from one single ancestor. So, in that case you have to use this kind of unrooted tree. So, depending upon your data, whether your data is a diverse kind of data, whether it is a very similar kind of data, whether these are the isolates of the same things and not. So, those kind of things if you want to analyze, you have to use this type of things. So, assume here taxa, all the taxa derived from a common ancestor, and in this case you assume that all the taxa do not have a common ancestor. So, depending upon your data set, you have to present your tree as rooted tree or unrooted tree. Next. So, the development of tree. Thank you. So, method of development of phylogenetic tree, there are two different methods of development of phylogenetic tree. One is called character based method, another one is called distance based method. So, the methodology by which you develop the tree, another most important thing, that you have two different methods, character based and the distance based. In the character based method, what happens, that suppose these are the two sequences. Now, these two sequences are highly similar sequence, the isolates of the same thing, that means you expect that they are highly similar sequence. So, if you are expecting they are highly similar sequence, that means they have very few mismatch or dissimilar sequence. So, in that case you only calculate those dissimilar sequence, that among these only one nucleotide is not matching. So, from that you give a score 1 because other sequence is matching, and then develop a matrix and develop a tree. Okay. The opposite thing, you are handling diverse sequence, very diverse sequence. Okay. So, in that case you expect that few nucleotides should match, maximum will not match, few nucleotides should match. So, again you score those few nucleotides which are matching. In this case you score those nucleotides which are not matching if they are very similar. So, in both the cases you are actually analyzing the characters or the nucleotides which are different. Whether it is highly similar match, then you find out the different sequence, the mismatch sequence. If they are highly diverse, you try to find out the highly similar sequences. Okay. So, in both the cases you are only seeing the nucleotides. Okay. So, characters you are seeing. So, that is why it is called character based matching or development of the tree. Now, there is a distance based method. Okay. Suppose you are analyzing these two sequences, there is T and G, you are taking another sequence where it is again T. Now, this T you have go back, again now come. Okay, so T and G and here it is T. So, what happens, you may think that this T and this T is same. So, next, so there is score 0 because they are same, but in this case their score is 1, but it may be happen next, instead of T earlier it was G, and in the next generation due to the mutation it again revert back to the back mutation and it revert back to the T next. So, what happens next? So, here score is 0, but there is a genetical distance. Okay. So, we are not taking care, in case of character based method, we are not taking care of that distance aspect of the sequence, but in this distance based method we are taking care of that distance also, that genetical distance how much, and through this we create a distance matrix and develop a tree. Okay. So, there are two methods, character based method and distance based method. Next. So, the example of character based method which we use very frequently, the first one is called maximum parsimony method. In the software you will find out this kind of options that create phylogenetic tree or algorithm, use the algorithm maximum parsimony. So, maximum parsimony method you use when your sequences are quite similar and number of sequences are small. Okay. You use this DNA parsimony, maximum parsimony. Okay. So, if you are handling different isolates or different strains of the same insect, then you should use only maximum parsimony. Okay. No other method, because you are handling similar kind of sequence. So, maximum sequence will be same, only few nucleotide will be different, based on those different nucleotides your phylogenetic tree will be developed. The opposite thing is called maximum likelihood. In this case your sequence is highly diverse, and based on those diverse sequence only few nucleotides will be matching. Suppose you are analyzing whitefly, aphid, any other insects. So, in those cases different things you are taking, definitely you expect that they will not be very highly similar. Okay. But only few nucleotide may be matching. So, based on those matching sequences they will generate the tree. So, either you have a very highly similar sequence or not very diverse sequence. In both these cases you use character based approach. Okay. Or either maximum parsimony or maximum likelihood. Next. So, example I am giving, suppose I am taking a human, a rabbit and chicken. I am trying to analyze some sequence. So, this sequence is showing that in human and rabbit in this gene there is only one mismatch. So, score is 1. In human and chicken there are 3 mismatch, score is 3, and rabbit and chicken there are 3 mismatch, score is 3. So, we can develop a matrix. So, this is called matrix. So, between human and rabbit 1, human and chicken 3, rabbit and chicken 3. Okay. So, this is called matrix. So, software will generate this kind of matrix, and from this matrix then what happens? So, the lowest score is 1, that means they are closely related. The lowest score is 1, that means they are closely related. So, you take human and rabbit 0.5 and 0.5, you join them together, and then the next one is coming chicken with human 3, rabbit with chicken also 3. So, what will happen? The chicken will come like this. So, this is 1.5, 1 and 0.5, that means 1.5 and 1.53. Here is also 1.5 and 1.53. So, in that way you develop a phylogenetic tree. So, tree is developed like this, software develops the tree like that. First it takes the smallest one and then build up the sequence on based on that. Okay. Now coming into the distance based method, the one example is the method is called neighbor joining method. So, neighbor joining method, it creates distance matrix. So, distance matrix means it creates a matrix which shows how much distantly it is related. So, the low value means they are closely related, high value means they are distantly related. So, here I am showing the relationship between spinach, rice, mosquito, monkey, human. So, different things. So, when you may think it as whitefly, aphid, some other things like that. So, different things we are taking and we try to analyze the sequence. So, the software will generate a kind of distance matrix like that, and here the lowest value is coming between monkey and human, the 3. Next. So, this is the lowest value. Next. So, what software will do? First step, they create a group monkey human creating these 2 branches. So, these 2 branches monkey and human, and this is the group. Now, monkey human as a group, the software will analyze monkey human as a group to find out the similar or distance between all these 3. So, next, in the next cycle monkey human as a group it will take, and remaining 3 they will analyze, and here it is the lowest value between monkey human with mosquito as a group with mosquito, the lowest value. That means this is the closest to monkey human. So, next. So, what will happen? Mosquito will come and this will come, it will form a group mosquito, monkey human and mosquito as a group. So, in that way tree is building up. Now, it will try to find out the mosquito monkey human as a group with spinach and rice. Next. So, in the next cycle when the software gives, it is not give with the mosquito monkey with any of them, rather it is giving the lowest value with spinach and rice. It is not giving with mosquito human, rather it is giving with these 2. So, that means in this phylogenetic tree there is not a single origin, there are 2 different origin, and when in a phylogenetic tree you will see when you analyze your sequence there are different clades, one clade together and another clade totally different. So, in those cases if it is like that, then we call them as polyphyletic origin. It is not a monophyletic origin that from one branch everything is going on and going on. It is not a monophyletic thing. But if you take a kind of similar isolate of one particular insect, in that case it is everything is monophyletic origin, but in this case it is not monophyletic origin. What happens next? So, rice and spinach will form a separate clade, and the last step is that to find out the relationship between spinach rice with mosquito monkey. So, what will happen? Now, it will form the entire tree. There are 2 clade, one belongs to rice spinach because they are originating separately with mosquito monkey and human separately, okay? And this is the group. But if you think that whether this clade and this clade holding together always, so if you do like that, then you will see if you do a bootstrap analysis, that time you will be able to know that whether their relationship to hold together is how much confident it is there. Next. So, the bootstrapping is the important thing. What is bootstrapping? The bootstrapping is the method of assessing the statistical significance, the position of branches of a phylogenetic tree, how much significant it is. So, to tell it in a very lighter way, whenever a boy and girl wants to marry, in many of our Hindu religion, there is a concept of matching the horoscopes. Okay. Why? This is the kind of bootstrap matching. So, by taking different type of analysis, it will tell that after marriage whether this boy and girl can stay together in a house or not. if any boy will come, whether the girl will deviate with that boy, or if any boy will come, the girl will deviate with that boy. So, that kind of things actually the horoscope is doing. Similarly, the bootstrap is doing the same thing. If any new sequence will come, now with the analysis it is showing that A and B isolate holding together. Now, if a C isolate or Z isolate will come, whether A form pair with Z or no, A and B always holding together, how much is their means confidence to stay together. So, that analysis is done by bootstrapping. So, it is a statistical analysis. What it does next? It does like that if these are the different sequence. So, 1 to 10 suppose I am showing here. So, it do the replication. So, in this after 1 it takes 5, then 6, then 2, then 3, that means this nucleotides positions they change, the software change those nucleotide position randomly, and then again they try to develop the tree. So, in from this replication they develop the tree like sequence A, B, C comes like that, sequence A, C, B comes like that. That means with this A and C comes together, with this A and B comes together, with this A and B again come together. So, out of this 3 replication 2 is showing the same thing. So, consensus is 2 is same. So, that means percentage if you calculate 75% is there the chance that these 2 sequences to hold together. Okay. So, similarly software will analyze by changing those nucleotide position randomly to know that whether these sequences are holding together or not. If they are holding always together, then the bootstrap value will be high. If the sequences are not holding together, bootstrap value will be less. So, from that value we will be able to understand whether this group which you are creating through the phylogenetic tree, how much is the significance of this phylogenetic tree. Next. So, there should not be any confusion of this phylogenetic tree development. First you choose a set of related sequence, obtain a multiple alignment, ask that whether there is a strong sequence similarity. You ask yourself by looking at the data. If that is there, yes, it is 95-98% similarity is there, then do a maximum parsimony tree and then do the bootstrap and develop the tree. If the answer is no, there is no strong similarity, then ask the question, is there any clearly recognizable sequence similarity? Whether the sequence similarity it may not be 95-98%, but whether it is 60%, 70% or it is 20%, 10%. What kind of, whether it is a recognizable sequence similarity, if it is yes, then do a distance based method like neighborhood joining method, develop the tree through bootstrapping. If it is no, that means there is no clear recognizable sequence similarity, which indicates the sequence has highly diverse sequence. In that case, you do maximum likelihood method and develop the bootstrap value and develop the phylogenetic tree. So, through all these things I think now it is clear that whatever is your data set, depending upon that you have to instruct the software what kind of phylogenetic tree you want. It is not that software will tell you whether you will develop a phylogram, whether you use a maximum parsimony, that you have to tell the computer what kind of things you want. Next. So, there are limitations of this bioinformatics, because bioinformatics can only predict, it is not always 100% correct. It many times it gives erratic result. As different subjects are interlinked to each other, understanding is very much difficult, and it may be possible to make relationships or model, but in reality that may not be correct. So, starting from the first I told you that we the civilized creature of the mother nature, which mother nature took billions of years to develop us, and we are trying to take the challenge with the mother nature to solve this mystery by just sitting on a computer with the softwares. So, let us see who will win, whether our mother nature or a silicon chip. Thank you.
Any queries from students?
Anything, any question, if anybody is working in this aspect, then it is very relevant to them.
regarding the bootstrapping. Regarding the bootstrapping, why would the software change the nucleotide sequences, like you said, like when the bootstrap is going on, shuffling the nucleotide sequences?
Because
I couldn't get it.
because whenever bootstrapping means that if the a new sequence, if it has to come, how it will come new sequence? New sequence come through mutation. From the same sequence few nucleotide may change. So what software will do, the string of sequence which is there, they take the 5th nucleotide to the second nucleotide to the 8th, like that they create that kind of mutation arbitrarily and develop different types of again alignments. Same sequence they are taking, only they change the nucleotide position 1 or 2 places, and in that way 500 times, 600 times they do different combinations, and with this combination they again generate the phylogenetic tree. To know that if any mutated sequence will come, a new sequence, mutated sequence means new sequence. So, in that case also whether this probability of holding these branches are correct or not, because everything the software is analyzing through prediction. So, they are trying to see that if any change happens in those sequences, then also whether it is holding true, because it is not doing all the nucleotide change at a time. In one sequence 1 or 2 nucleotide it will change, next sequence again another 1 or 2 nucleotide it will change. So, in that way it generates different sequence and again do the multiple alignment, again develop the phylogenetic tree. It is not that the entire sequence it has changed, only few nucleotide it has changed, so that it seems that a new sequence has come. So, in that case whether they again hold together or not.
Next is, sir, regarding this, could you please tell in bit more details regarding the character based and the distance based methods of the development of the phylogenetic tree?
Details means what? Means if more details you require, I am always available in this building. Practical sessions when we will do, that time also we will see, and if any details is there, requirement is there, you will be here for next 10 days. So, I am just sitting in this lobby only, second room. So, you can come and we can discuss in details, but for your practical things only these two terminologies is important, and when you have to use those things, that is important, because if you go in depth of the statistical part of that thing or algorithm part of that thing, it will be very much not very much interesting for a biologist to know what is the algorithm part of the distance based matrix or character based matrix.
Sir, regarding the data mining, so regarding the different databases, like we are going for the mostly for the NCBI or the other databases, we are getting same sequences from that. Suppose if we have any new gene, we have to mine for a new gene, then do we have to look for the different databases and to get this predicted sequences from those databases, then to like in the process of choosing a gene?
Means you want to know actually that allele mining. So, if you suppose if you have a genome sequence, that genome sequence not always deposited as a full annotated sequence, only few genes which is already known, those sequences has been annotated and deposited as a raw sequence in the database. What people used to do, take out those sequence again, do the unannotated sequence, because there are many hypothetical protein sequence has been deposited. You take those sequence again, do much more analysis, not only through the NCBI, there are many other software. So, one by one you have to go, you have to first analyze the secondary structure of the protein, quaternary tertiary structure of the protein, identify the super secondary structure like motifs, like domains you have to identify. Then if those things are available, then there are other softwares are available to build up a 3D model of the protein. So, if those things are there, then you can find out a sequence and their means corresponding protein sequence. But all these things again I am telling, they will predict. You have to do their analysis in the lab.
If there are no questions, I profusely thank Dr. Anirban sir for his wonderful lecture on the molecular phylogeny principles, concepts and practices. So I request our course director to present a moment on
Excellent. With this, we conclude this first part of this program. Afternoon, we'll be assembling at 2:30.